So good afternoon, everyone. I'm very happy to be here. My first time today in Receptor 2025. And at the outset, I would like to thank Dr. Kumruel sir for giving me this opportunity. I think we are almost at the fag end of the narrow window as told by Dr. Arun of the postprandial state. I think everybody is about to start napping, so I'll try to make it as crisp as possible. So my topic today here will be about the gut microbiome and its linkage with diabetes and the various research and clinical applications. So to begin with, what is gut microbiome? We interchangeably use the term that is gut microbiome and microbiota. So the microbiome is nothing but the microbial community, its genes and the metabolic products generated by these bacterial holders, which inhabit the entire length of our human intestine. Whereas when we refer to the gut microbiota, we refer only to the microorganism per se and not to the genetics or the metabolic products. So actually the gut microbiota outweigh our cells. So each one of us consists of approximately 40 trillion human cells, but the human gut hosts about 100 trillion microbial cells with an average weight of around 1.5 kilograms. And it changes, it is very much diverse. It changes across the spectrum of life from infancy to adulthood. It changes from person to person. It has regional and ethnic variations as well. And in spite of all these variations, the majority of gut microbiome belong to these three particular species, that is firmicutes, bacteroids, and actinobacteria. So because of this particular reason, any imbalance in the first two bacterial species are going to be the causative factor for majority of diseases. So when we go back to the history of this gut microbiome, it's very interesting to see that Hippocrates, as early as 460 BC, has talked about the beginning of the disease in the gut, and the health is determined by the microbiota in the gut. And even our Ayurveda talks about the toxic products that are present in the intestine, which lead on to various diseases. So as early as 1600, the very first microbiologist who started identifying microorganisms through a lens was very surprised to hear that he was actually a shopkeeper from Netherlands who identified these lenses which could magnify microorganisms, so magnify things to 270 times and identify these microorganisms. And he started identifying these as he called as animalcules, that is minuscule animals as he called them. And then this was the first origin of microbiome as well. And then later from France, Professor Ilya Mechnikov, he was the one who first identified that lactobacillus is a very good species of bacteria which can cure diarrheal diseases in children, and this led to the invention of the first probiotic. So we are talking about so much trillions of bacteria, so now we get a doubt whether you're good, whether you're bad. As Kamal says, like the kid asks Kamal Hassan, "Ninga nallavara, kettavara." So we have this doubt. Now we are talking about the correlation of gut microbiome in relation to metabolic diseases as well, beyond the role of infectious diseases caused by this microbiome. So very natural that we get into this doubt. So the gut microbiota composition, as I told you, it changes from across the length of the intestine, and the composition also goes increasing as we cross from the duodenum towards the colon. And the majority of the microbes that live in the small intestine are facultative anaerobes, whereas the one in colon are a very, very strict form of anaerobes. So what are the factors that impact gut microbiome? So there are a lot of factors, right from our own concept of like mode of delivery, whether a normal vaginal delivery versus cesarean section, which decides the microbiota inhabiting the baby's intestine. So right from there, the genetic makeup of the host, the delivery, the duration of lactation, the introduction time period of complementary foods for weaning, the geography, the antibiotic usage, the dietary pattern, and so on. So many things impact the gut microbiome present in a single individual person. So a very good thing. So what happens between these two, to the human being and the microorganism? What do they gain from each other? So they live in a constant state of symbiosis. The microorganism gets a shelter, nutrition, and it grows as well. Whereas compared to this very, very minimal amount of benefits it reaps, it magnifies and gives multitude of benefits to the host. As you can see here, it controls inflammatory and immune responses. It is responsible for the maintenance of intestinal barrier integrity so that it prevents toxic substances entering the human circulation. And it leads on to help with production of vitamins and proteins, helps with bile acid metabolism, which has a very huge role to play in immunity and metabolic diseases, and also it has a role to play with metabolism of drugs. So what happens when this gut microbiota goes haywire? So this is called as dysbiosis, abnormal microbial colonization of the intestine, which leads on to harmful problems. This can happen with the changes in bacterial composition or a decrease in abundance and diversity of the various individual groups of species. So these are the association with various diseases, right from heart disease, cancers, Parkinson's and Alzheimer's, prostate cancer, allergy and asthma, right now to obesity and diabetes, and also to autism and osteoporosis. So what not, the list is actually very, very huge. So now coming on to the topic per se, what is the link between the gut microbiome and diabetes? So initially when the scientists had a human microbiome project based on metagenomics in 2012. So they tried to study the genetic makeup of all these microbiomes. They first try to identify the species and then they try to identify the genetic structure. So when they started doing this, they were able to identify in different groups of humans and different disease and health states that the group of species were altered. There was a preferential increase in some of the bacteria in some disease states and in contrast to the normal person. So that is how this link was discovered between the gut microbiome and obesity and type 1 and type 2 diabetes. So analyzing bacterial gene functions reported a discrepancy in the gut flora composition at the molecular level. So going on to the link with type 1 diabetes, as all we know that the type 1 diabetes prevalence is recently increasing, and the recent association with gut microbiome is slowly being established very well. So this 16S pyrosequencing is a methodology by how they identify the various species of the gut microbiome. So what they found that even before the development of type 1 diabetes, that children who had a positive islet and autoantibodies had a higher prevalence, higher ratio of bacteroids to firmicutes and a lower gut microbial diversity. So this is what they found. These are the species that are altered, and this is what happens because of that. So going into the mechanism proper, so what happens is there's an increased intestinal permeability happening because of endotoxins, and there's a decrease in gut microbiome diversity. I'll come on to this later. There is something called as butyrate producing bacteria. Whenever the number of this bacteria goes down, it leads on to a lot of inflammatory processes. So what happens as the result of this is there are a lot of autoreactive antigens which are entering the circulation, which can activate the innate and the adaptive immune system, so which leads on to autoreactive T helper cells which can produce insulitis or beta cell destruction. The same way can happen a lot of macrophages and adaptive immune system again going on to cause beta cell destruction. And not only that, some of the metabolic products produced by this harmful gut microbiome can produce a state of molecular mimicry. They can mimic like an endogenous antigen, again producing pancreatic beta cell damage. So this is what happens, altered microbiota, abnormal epithelial activation, there's lot of cytokine released which leads on to T and B cell activation, leads on to pancreatic inflammation and beta cell destruction. So coming on to the link with type 2 diabetes. See, type 2 diabetes, the pathophysiology mechanisms have grown by leaps and bounds. We started with just 2 mechanisms, and then for many years it was just de Franco's ominous octet. Now we have gone on to more than 18 and 20, up to 20 mechanisms which have been found to be associated with diabetes. So this is one of the concepts that is egregious leaven. along with the usual concepts, what we know about the association with causation of diabetes, there's also an association of gut microbiome leading on to abnormal incretin production and insulin resistance leading on to diabetes. So these are the alterations of gut flora in type 2 diabetes individuals. So this is what I was talking about. So this state is actually uniform in both type 1 and type 2. That is a decrease in firmicutes and increase in bacteroids. So when we talk about, though the bacterial alteration is the same, but the pathways leading on to type 1 and type 2 diverge from this point. So we talk about two things, that is storage versus inflammatory hypothesis, which can lead on to type 2 diabetes. The storage hypothesis is nothing but, so excessive consumption of high-fat foods can lead on to modulation of gut microbiota, which leads to decreased butyrate-producing species. Because of this decreased butyrate-producing species, other harmful microorganisms grow, which leads on to increased production of endotoxins. So this is called as leaky gut hypothesis or metabolic endotoxemia, so which can lead on to inflammation and lead on to the various pathways associated with insulin resistance. So these are the other mechanisms. Along with the increase in lipopolysaccharides, there are changes in bile acid metabolism, alterations in short-chain fatty acid production, alteration in GLP-1 production from the L cells of the small intestine, and changes in circulating branch-chain amino acids. So this is what I just told you about the increased prevalence of lipopolysaccharides causing increased gut permeability. So this is one thing which has to be dealt, because this is the basic of probiotic and prebiotic administration in diabetic individuals these days. So in colon, the bacterial fermentation leads on to production of acetate, propionate, and butyrate. So acetate is an energy source for the colonocytes, whereas butyrate and propionate are associated with various metabolic functions. So they can control the production of GLP-1 from the small intestine. They can directly, through their mechanism on bile acids, they can directly control the hepatic metabolism, and as well as in the muscles and adipose tissue, they can control the AMP kinase and lipolysis activities as well. So this is in summary. So these are the various mechanisms which can be causative towards type 2 diabetes. So what are the clinical applications? Now we have understood the link between type 1 and type 2 diabetes. So what is the link? So we keep thinking of these gut microbiota as very little small devils, but they are not so. The modulation of this gut microbiome in a very, very beneficial manner through all these processes like diet, exercise, anti-diabetic drugs themselves, pre versus probiotics, and finally fecal microbiota transplant has a very long way in producing remission of diabetes and achieving a proper glycemic control. So very surprisingly, Western diet with a very, very little amount of fiber and high saturated fatty acids produce high amount of endotoxins leading to inflammation. So this is what happens in the gut microbiome when you consume lot of saturated fats diet. So when you go on with Mediterranean diet, it produces increased amount of butyrate, which I already alluded to, which is very protective in terms of type 2 diabetes. So this is what we'll be able to offer in future. We can offer personalized, tailor-made diet strategy based on the composition of our gut microbiome. So this is otherwise called as precision nutrition, which is coming up in a big way. So even exercise, so high intensity exercise, in contrast to our original belief, actually causes dysbiosis in the gut microbiota, and it can produce gastrointestinal syndrome. Whereas moderate intensity exercise actually increases the beneficial species like Akkermansia and butyrate producers, which leads on to beneficial aspects. So now, coming on to anti-diabetic drugs, we have always been believing that anti-diabetic drugs act through the various pathophysiological mechanisms alluded to in the ominous octet, but not so. Now we have clinical evidence that even anti-diabetic drugs act through this modulation of gut microbiome, which helps us to reap a lot of potential benefits. And what they say is the adverse effects what we develop to anti-diabetic drugs, for example, the GI intolerance that develops to metformin very commonly, even that is dependent on the composition of a gut microbiome of a person. So the metformin has been extensively studied in this regard. So metformin causes decrease in bacterial diversity, increase in butyrate producers and Akkermansia species, and it reduces the inflammatory status present in the gut. And this is what I talked about. Increased presence of Streptococcus parasanguinus is predictive of metformin-associated GI intolerance. And not only metformin, the other drugs as well. Gliptins have been shown to increase bacteroids. Liraglutide increase short-chain fatty acid producers. SGLT2 inhibitors, it's still new kid on the block, so the effects on gut microbiome are contradictory, no conclusive evidence. And alpha-glucosidase inhibitors, again, they have a lot of beneficial effects. So coming on to the pre and probiotics, as we know, prebiotics, they are complex carbohydrates, polyphenols, and polyunsaturated fatty acids like oligofructose, capsaicin, barberin, etc. Again, these ones act in a similar way, increase in GLP, short-chain fatty acids, etc. And probiotics, again, do they improve glycemic control? So though these are live microorganisms, lactobacillus, bifidobacterium strains, and Saccharomyces, which is commonly used in children for diarrheal diseases, so they can increase the Akkermansia mucinifera species, which helps to regenerate the intestinal barrier and improve the metabolic processes. But to remember, pre and prebiotic combination, that the symbiosis is only adjuvant therapy, not curative on its own. So these are the studies in that regard. So coming on to the last thing, that's the fecal microbiota transplantation. So this is nothing but transfer of stools from a healthy donor into another subject gastrointestinal tract by either capsules or by an endoscopic formulation. So this is usually, it can be an allogenic or an autogenic, allogenic from a donor or an autogenic from the self itself. It is cultured outside, the microbiome is altered and again given back to the person. See, we have all known about this FMT in terms of Clostridium difficile infection and inflammatory bowel disease. But in relation to diabetes, these days a lot of studies are coming up at a big level telling us the efficacy. So it alters the natural course of even type 1 diabetes, and hence a lot of studies are going on in pre-type 1 diabetes and type 1 diabetes, whether alteration of gut microbiome can modulate autoimmunity before the disease onset happens and whether there is a decrease in autoimmunity because of that. The problems are, the caveats, the success dependent on the tested strains, the composition and diversity, and it's very funnily, an article states that one stool does not fit all, which means because of the variation in genetic makeup, the preparation of FMT might not fit every person, so it needs to be grown even more by research. And the risk, that is transfer of infectious disease or a promotion of dysbiotic status if proper screening is not done. So this is what the studies in diabetes say. They compared FMT with FMT plus metformin. So patients who had effective engraftment, so they had achieved a very good glycemic control and improvement in metabolic parameters in terms of insulin resistance. So these are the beneficial bacteria which went up. So how do you identify? You're talking about trillions of cells. Now you come to a state like you want to know the diagnostic test as a clinician. How do you want to identify? Initially, it was culture-based, which is very, very difficult to identify the species. And now there are a lot of next-gen sequencing tests, especially the 16S rRNA is very, very particularly specific for bacteria, which is present in the gut. And the metagenomics, the combination of the biomarker sequencing and the genetic studies. So these are all the various tests that are applied. Still not available to the common person, still available at research labs at a higher level. So this is about the metagenomics. So they isolate the fecal sample, they break down the DNA and they sequence it and analyze it. So now, so we have talked about the link between the gut microbiome and diabetes and what all the treatments and applications that can be offered. But still we are a very, very long way from achieving all this in our day-to-day practice because majority of microbiome is not cultivable. Because we are not only talking about bacteria, we are also talking about virus and protozoa, which are very, very difficult to culture. And the methodology of metagenomics cannot be applicable to everyone and cannot be available everywhere. And also long-term prospective cohort studies has to be available to define the causative process. So these are the various things which are happening in the world of gut microbiota, along with fecal transplant, something called as ecological engineering, and medications which can only target the microbes which can alter the metabolic pathways and target the host microbe associative pathways as well. So these are some things which are excitingly happening in the world of gut microbiome research. So day is not far when doctors do gut profiling and we can give food plans to patient accordingly. So these are the take-home messages. So there's an altered gut microbiome in both type 1 and type 2, and these are the bacterial alteration. Primarily, there's a decrease in butyrate producers, and there's a bidirectional relationship even with anti-diabetic medications and gut microbiome. So fecal microbiota transplantation is coming up in a big way for this association. So thank you.